see. Hi everyone, I'm Nidhi and I'm from Jake Grossman's lab, and I'm excited to present to you my summer project, which has been maple hairstyles across the globe. Okay, so today I'm going to talk about journey of maples, a little about leaf hairstyles, trichome, a few of my research findings, and then we'll have time for questions and answers at the end of 7 presentations today. Okay, so I chose maples as my study species because Jake has been working on it for a long time, but I'm also really fascinated by their color and abundance in temperate region. I thought it was also apt to choose maples because the DaRin Arnold Arboretum is within the Maple Nations, a collection of indigenous people that have been stewarding care and conservation of maples for many generations now. So the journey of maples looks really confusing over here, but if we look at the map on the left and focus on the yellow and red colors and compare it to the phylogenetic map on the right, we can understand that maples originated 75 million years ago in East Asia and radiated towards North America, Europe and North America over time. And therefore, you see a large diversity of maples on the eastern part of the world compared to the western. And also, you see that many of the traits, like leaf size and fruit, are phylogenetically related, but we know very little about leaf hair. And these are some of the photos that Jake and I took in the lab downstairs. So why leaf hair, and what is leaf hair? Botanically, leaf hair are called trichomes. When we take a leaf, this is a paper bark maple, and look at it under the microscope, we see the presence of trichome on leaf surface. The presence of hair, this is taken by Faye and as interns with SEM. So the presence of hair in many species has been documented to influence leaf functions like photosynthesis, drought and heat tolerance, warding off predators, but we know very little about this when it comes to maples. So therefore, I asked 3 main questions to further our understanding of leaf hair. My first and most basic question being, how many maple species have trichomes? To answer this question, I used data from publicly available guides and then categorized 160 species into pubescent, which is presence of hair, absence, which is glabrous. From this, I got a pie chart, a simple pie chart that gives us the information that 80 species and 51% of maples are pubescent, 49 species, 31% are glabrous, and we don't have information for 29 and 18% of maples. Then I further wanted to understand if there's information about if the hair that is present on the species are on the top of the leaf or on the bottom of the leaf. So with further categorization, we can see that 37% or 58 species have hair predominantly on the bottom side than on the top side, which has been documented to be just about 1% for 160 species. And about 20 species and 13% have hair on both sides. So my next step would be to understand if the case is that a lot of species have hair on the bottom side, and why this might be. We hypothesize that this might be because of stomata being predominantly on the lower side, which conserves water. So that will be one of my next steps. My second question was, is the presence of trichomes phylogenetically conserved? To answer this question, I used Arasis et al.'s phylogenetic map. And on the right, you see all these species that are grouped into sections. So what I did is I categorized or labeled all the species that were pubescent, that were glabrous, and then information that was not available. From this, we see an emerging pattern where certain sections are more glabrous, whereas other sections are more pubescent. Using statistical analysis and Pearson's chi-square test, we see that there is a significant value, but this does not entirely mean that this is significant because there's a lot of data that is missing over here. This is categorical. And so my next steps would be to create exactly the same with leaf hair density. My third and last question was if bioregion and climate predicts the presence of trichomes across species. To answer this, I categorized the data that I had into 5 main categories based on where they're present in the world. And then using R and Pearson's chi-square test again, we see a significant value, but much like the earlier slide, this is because trichomes are not equally distributed across the bioregion. And finally, we were curious if global climate, like hot, precipitation, and cold, predicts the presence of hair. To answer this, I used Jake's data that gives us information of a subset of species that is sparsely distributed across the globe, but gives us information about their native temperature and mean annual precipitation range. So using logistic analysis, I got the 2 plots that basically show that mean annual temperature and mean annual precipitation give us very little evidence of the presence of hair so far. So my next key steps would be to revisit all the 3 questions, but to first collect observational data and update the information that we have been using for many years, and then finally, to use leaf hair density as another measurement that would quantify a lot of the questions I presented. This is important because 22.8% of the species are threatened, although 71.5% are not threatened. And with climate change, we see that we don't know the information for 106 species. Their population trend remains unknown, and 32 species show decreasing trend and 20 are stable. So understanding leaf hair would show what species are more tolerant to drought and heat stress and what species might not be. So it will open a lot more questions. Finally, I want to say that I'm not the only one studying leaf hair. There's a haircut company called Maple Leaf Hair, and many people are excited about having maples on their hair. And lastly, I want to say thank you, Faye. Thank you, Jake, for so much support and teaching me a lot. Thank you for all the interns, and also a shout out to Ellie Mendelson, who helped me clear all these leaves to do further analysis. And lastly, I want to say that last year I was in a really hard situation as an international student, but Arnold Arboretum really helped me get to the place that I am today this summer. So thank you all.
Thank you, Nidhi. Next up, we have Dylan, who has also learned Jake Grossman's lab.
All right. Hi, everyone. My name is Dylan. I am from Jake Grossman's lab, and this summer my research focused on how warming spring temperatures and other plant traits influence the leaf out phenology for woody species at the Goucher College Arboretum. So phenology is the study of reoccurring biological events, and more specifically, leaf out phenology is the study of when species leaf out in the spring and what cues are received by plants in order to leaf out. The 2 most important factors for leafing out are photoperiod and temperature. And in temperature, the 2 most influential roles are winter chilling and forcing temperatures, which are just the warm spring temperatures that allow the bud to get to a point of bursting, otherwise known as bud break. So how can leaf out phenology be used to investigate the impacts of climate change? Well, since there's such a strong relationship between temperature and leafing out, researchers over the past couple of decades have spent a lot of time trying to understand how warming temperatures influence phenological patterns. Botanical gardens and arboreta are very important for conducting phenological research because they have a wide array of species from all around the world, as well as meticulous records that allow us to understand how phenological patterns have shifted. At Goucher College, I have been working to help establish an arboretum, which I will be using to collect phenological data for my senior thesis. So the questions that I asked this summer were, how are leaf out times for hardwood species in Goucher's living collection shifting with changes in average springtime temperature? What qualities of hardwood species known to influence leaf out date influence the order of leaf out timing? And how can the Goucher Arboretum be utilized to investigate changes in leaf out phenology? While I won't go into my methods too extensively, I collected my bud break phenology data from the National Phenology Network, and I collected the associated climatic variables from the Climate NA map. And all of my statistical analyses were done in RStudio. And if you have any other questions about my methods, I'd love to take those later this afternoon. All right, so in order to answer my first question, I ran linear regression models comparing the average spring temperature to leaf out day for 16 species in the Goucher College Arboretum. For Acer rubrum, red maple, I found there was a negative correlation between these two variables. So as average spring temperature increased, the leaf out day advanced. So for all of these linear regression models, they all had a negative slope, but not all species fit the linear regression model equally. So you can see in the first two rows in red and green, their average spring temperature definitely had an influence on leaf out. But all of the factors that influence leaf out day were not included in the model. So there must be further research to exactly understand what influences leaf out day for these species. Down in the last row in orange, you can see that the adjusted R-squared values for these models were below 0.1, suggesting that the relationship between the two variables was not that strong, and further research needs to be done to understand what influences leaf out day for those species. Now for Atilia americana, American basswood, you can see the linear model did not fit as well as a nonlinear model, which suggests, again, there's a nonlinear relationship between these two variables. And for Pappus deltoides, eastern cottonwood, I actually found that there was a positive correlation between the two variables. So as average spring temperature increased, the leaf out date also increased. And this is supported by research with two closely related species, quaking poplar and big-tooth aspen, that showed that the leaf out day was actually much later when the days of winter experienced were shorter, suggesting that these species require a stronger winter chilling requirement in order to leaf out earlier in the spring. In order to understand how the evolutionary history of species influenced the leaf out date, I created a phylogeny for the hardwood species at Goucher, and using the average leaf out date from the Panchin et al. 2014 article, I was able to test for phylogenetic signal in the phylogeny. So phylogenetic signal is the tendency for closely related species to resemble each other more than a species chosen at random on the tree. And so, in other words, I was looking to see if closely related species retained or conserved their leaf out day, or if this leaf out day is randomly distributed throughout the phylogeny. So the associated Bloomberg K value is 0.168, which suggests that there is weak phylogenetic signal in the phylogeny. And using the Lipa-Moran function in phylosignal, I was able to actually visualize the signal in the phylogeny. And I found that clades with significant autocorrelation were Chamaecyparis, Aesculus, Ilex, and Aurelia, which suggests that they have conserved their leaf out day throughout their evolutionary history. So how does stem anatomy influence leaf out events? In order to answer this question, I compared the leaf out day between species with varying pit membrane sizes. So in the xylem tissue of hardwood species, water and nutrients are transferred from one vessel to another through bordered pits. And bordered pits also prevent gas from entering the vascular system of plants, which is known as embolism. Past research has suggested that the degree to which a plant is susceptible to embolism influences when they leaf out. And pit membrane thickness has been shown to have a very strong correlation to this resistance to embolism. So I hypothesized that species with smaller pit membranes and consequently larger pores would leaf out earlier in the spring because it would take them less time to receive those water and nutrients, with the trade-off that they are more susceptible to embolism. And in the contrary, species with larger pit membrane thicknesses and consequently smaller pores would leaf out later because it would take them longer to receive water and nutrients, with the trade-off that they are less susceptible to embolism. So using a data set of 16 species, which were different than the 16 species I used for the linear regression models, I was able to find that pit membrane size had a positive correlation with average leaf out day. So as the pit membrane size was larger, the later the species leafed out. All right, so for next steps, I'm going to be continuing my phenological research at Goucher College. On the left is a map of at least one of every hardwood species, along with the scientific name and a photo of the specimen. And I also have a table which I'll use to collect different phenological phases, as well as some of the categories for the leaf senescence data collection. I plan on continuing to use PhyloSignal, the R package, to understand how the evolutionary history of these species influenced leaf out date and other traits, as well as understanding how temperature, precipitation, and elevation influence leaf out date. So I'd just like to finish off by acknowledging some people that made this entire experience what it was. Faye has been so kind. Jake, thank you so much for being a wonderful mentor. My advisor at Goucher College, Daniela Beal. And I'd like to thank the Arnold Arboretum of Harvard University, as well as the DaRin Butz Foundation. And lastly, the rest of my cohort who have been so kind, and I've just been so grateful to get to know. So thank you all for listening.
Thank you, Dylan. Next up, we have Yanni, and he's been working in the Templar Lab at BU this summer.
Hi, everyone. My name is Yanni Pappas, and this summer I worked with Dr. Pamela Templar and Emma Conrad-Rooney looking at atmospheric deposition in urban environments, primarily focusing on nitrogen deposition, what that looks like in urban areas, and what that might mean for both ecosystem and human health. So to begin, human activities have significantly increased the amount of reactive nitrogen present in the biosphere over the past century. And this has been through changes in agriculture and increased fossil fuel combustion, which both lead to nitrogen emissions. And these gases go up in the atmosphere and can be brought back to Earth's surface through atmospheric deposition. And atmospheric nitrogen deposition can have various impacts on all types of ecosystems. At low levels, it can act as a fertilizer, but at elevated levels, it can have a wide array of detrimental impacts. So as humans have drastically altered the nitrogen cycle, it's crucial that we understand the degree and nature of nitrogen deposition on our environments to best ameliorate the impacts. And for my project this summer, we used data from the National Atmospheric Deposition Program. And the NADP is a cooperative program of about 250 sites throughout the United States. And most of these sites are located in rural areas, as cities tend to be deposition hotspots, and the program sought to obtain an understanding of the background levels of deposition in the United States. But in recent years, there's an increasing recognition of the importance of understanding urban deposition, and new urban sites have been added, such as at Weld Hill at the Arnold Arboretum, pictured here on the right, and at Boston University. And every NADP site is the same. It collects wet deposition precipitation and analyzes it for these elements and compounds. And through this project, we use the NADP data to understand the nature of atmospheric deposition in urban areas. And we had two main questions. Our first was, are urban areas deposition hotspots? And if they are, to what degree are they hotspots? And our second question was, how has urban deposition changed over time? Which deposition types are increasing, decreasing, or staying the same? And pictured here on the left, we're looking at total inorganic nitrogen wet deposition in 2018. And the red is indicating elevated amounts of deposition. And the background of this map represents the interpolation of deposition rates across the United States based on the data from the rural sites. And these open circles reflect the urban sites in the program in the actual measured deposition of those locations. So we want to start to think about, how do these actual measured deposition at urban sites differ from the background rural estimates to try to understand our first question. And it's important to note that we're looking at wet deposition. So areas that don't experience as much precipitation won't show up in this map. And also all our urban sites are located on the eastern half of the United States. But we started with that map. So that was the total nitrogen pictured here on the right. And we split it up into ammonium and nitrate deposition, as both have different sources and different impacts on the environment. And while they exhibit similar patterns, we can see that the distribution of deposition is a little different. And we not only looked at nitrogen for this, we looked at all the other deposition types that the NADP collects data for. And as we can see, other deposition types aren't as widespread as nitrogen, but can be quite elevated in more localized areas. But to look at our first question for our urban areas and nitrogen deposition hotspots, we're focusing on ammonium nitrate and total nitrogen. And we're looking at these urban sites and comparing them to the background rural estimate for those locations. So to do that, we created this bar graph. And here we're looking, we have the different deposition types on the x-axis, ammonium nitrate, total nitrogen. On the y, we're looking at the deposition amount. And we're looking at the average for the NADP urban sites in 2018. Again, we're only looking at wet deposition. And the light gray bar here is the interpolated background rural estimate for the urban sites. And then the dark gray here is what's actually being measured at the urban sites. And the blue is the difference between the measured and the estimate. And as we can see for ammonium nitrate and total nitrogen, the blue is positive, indicating that urban sites are experiencing elevated amounts of nitrogen deposition compared to the rural estimates. And this led support to the idea that urban areas are nitrogen deposition hotspots. And for our second question, we looked at wet deposition over time for NADP urban sites. So here, each graph is going to illustrate a different deposition type. So we have ammonium nitrate, total N. Each color is corresponding to a different urban site. And on the y-axis, we have the amount of the deposition, and then the x-axis is the years. And we have data all the way from the late 1970s. And we also drew trend lines on these maps. The black is overall trends for all urban sites, and specific colored trend lines correspond to specific urban sites. And any trend line that we're illustrating here has been found to be statistically significant. So as we can see for ammonium, it's been slightly increasing over the past 40 years. Nitrate has been decreasing, but these two opposing trends lead the total nitrogen to be slightly decreasing, kind of staying the same over the past 40 years. And we also looked at other deposition types too, including sulfate and H+, which both exhibited really impressive downward trends. And this is interested looking at it through the political context, in that the 1990 amendments to the Clean Air Act that addressed acid rain put regulations on the precursor pollutants of sulfate deposition and nitrate deposition. And this regulations led to a decrease in acid rain. And something like ammonium, which doesn't, its precursor pollutant isn't regulated federally in the United States, has been increasing and opposes the deposition types that the precursor pollutants are regulated. So it's interesting we start to look at this data and think about why this might be, and think about the regulations in place for the different deposition types. And we also looked at the other deposition types as well, but as you can see, the trends aren't as clear, but we can see that certain deposition types and areas are experiencing really high amounts compared to other areas, which we'd be interested to look into further. But to conclude, we found that urban NADP sites experience higher levels of nitrogen deposition than estimates from the background rural levels. So this supports the idea that urban areas are nitrogen deposition hotspots. And then we are also able to see some wet deposition trends and understand that in the regulatory context. And this research is really important because anything that's falling from the sky is landing on the leaves, it's going into the soil, into the waterways, and is affecting the biogeochemistry and the health of these ecosystems. And especially in urban areas where ecosystem and human health are so closely linked, it's crucial to understand the components and trends of urban atmospheric deposition so we can best safeguard environmental and human health. So thank you for everyone for listening. I just wanted to say a big thank you to the Arnold Arboretum and the DaRin Butz Foundation for giving me the opportunity to be here this summer and conduct this research. I want to say thank you to NADP for the data, support from the Harvard Forest RU program, of course, Faye, who is so amazing and kind throughout this whole summer and put together an amazing program for us interns. And finally, Pam and Emma, who were so welcoming me into their lab and provided me immense support and made this project possible. So thank you very much to them and to everyone.
Thank you, Yanni. Next up, we have Erica, and Erica has been working with Al this summer.
right. Hi everyone, my name is Erica and today I'm going to be sharing with you what I've been doing this summer, which is using cold hardiness to predict bud break in maples of the Harvard forest as part of Dr. Al Kowaleski's lab. So let's get right into it. What is bud break? Bud break is a phenological event and is typically deemed to be achieved when about 50% of the buds on an individual have burst. Here we have the two species that I have chosen to study this summer. We have Acer rubrum and Acer saccharum, the red maple and sugar maple. And I chose to study the type of bud that was going to burst first in each species. So that would be the floral bud for Acer rubrum and the vegetative or leaf bud for Acer saccharum. So let's talk a little bit about why bud break is important. First of all, bud break is very important for species distribution and carbon cycling. So for example, a tree species whose buds freeze off in the middle of winter won't be able to reproduce in that cooler climate and won't be able to leaf out, which is necessary for realizing a tree's carbon sequestration potential. If we think about floral bud break in particular, plant and pollinator interactions come to mind. So you'd want the floral buds to be bursting at the same time that the pollinator for that species is most prolific. And along with that, bud break is also really important for determining the timing and length of the growing season for a lot of our agricultural crops. So why did I choose these two species of maple? Well, first of all, both belong to the same genus and have very broad overlap in their ecological niches, which you can see in the species distribution here on the left. And so I thought it would be interesting to compare these two species. So the conventional method for predicting bud break utilizes the relationship between accumulating chill and growing degree days. So it's typically believed that as chill accumulates, the amount of growing degree days required for bud break declines. And we can see this in this figure from Wang et al., where you can think of heat requirement as analogous to growing degree days. So as the chilling units are increasing, the amount of growing degree days required for bud break declines. And then we can use this relationship and relate the growing degree days to the day of year that bud break occurs. But before I get into my model building, I just want to talk briefly about what is a growing degree day. So growing degree days, also abbreviated to GDDs, are a measurement of thermal time. And it's believed that when GDDs are accumulating, there is metabolic activity that contributes to the non-reversible growth of the bud. So we can't have negative GDDs because we can't subtract from the growth that has already happened in the bud. Calculating GDDs is very simple. All you do is take the air temperature and you subtract it from a given base temperature, which is usually 0, 5, or 10 degrees C. So I have a couple examples to show. In this first example, we have the air temperature at 15 degrees and the base temperature at 0. Subtracting the two, we find that 15 growing degree days have been accumulated. In the next example, we keep the air temperature the same, but raise the base temperature to 10 degrees C. Subtracting the two, we find that only 5 growing degree days have been accumulated. And lastly, if we cool the air temperature down to 5 degrees C, we keep the base temperature the same. Since 5 is less than 10, 0 growing degree days have been accumulated. However, since it's very unlikely that the buds reach a state of metabolic stasis once the air temperature drops below this base temperature, it might not be the best way to be predicting bud break. That brings me to cold hardiness, which describes the ability of a plant to survive low and cold temperatures. There's a lot of different factors that go into describing the cold hardiness of a species, and we can utilize the relationship between these factors to predict bud break. So that brings me to my question for this summer, which was, can we improve bud break predictions with models based on cold hardiness rather than growing degree days? In order to answer this question, I first had to prepare my phenology data, which came from the Harvard Forest Data Archive. The archive contained 30 years of phenology data, which I separated into calibration and validation data sets using a random number generator, and you can see the results of that here. The calibration data set was used to build the model, and the validation set was used to test the model. So then I got to building my conventional GDD model in a way that is very similar to what I described earlier. So I calculated the cumulative chill, And then I used a linear model to relate that accumulated chill to the observed growing degree days at bud break. Then with this model, I applied it to the validation set to predict bud break and analyze fitness. And so we can see the result of the growing degree day model here, with observed day of bud break on the x-axis and the predicted day of bud break on the y-axis. This black line represents a one-to-one relationship. So if the model was perfect, the dots would be falling directly on the line. And so what we can see here is that it's a decent model. There's decent correlation and it's doing what it's supposed to be doing. But we're seeing that the model is over predicting on average about 3 days. So the predicted day of bud break is about 3 days later than what is actually observed. So there's some room for improvement. That brings me to building my cold hardiness model. So there's 3 main factors that we can use to relate bud break to cold hardiness. One is the initial cold hardiness of the bud. The next is the cold hardiness of the bud at bud break. And the next is the effective rate of deacclimation. So here we have a figure from some of Al's previous work. The dots represent the average cold hardiness of Acer saccharum, the sugar maple, over the growing season. And the little density curve at the top shows the observed day of bud break in the fields. And the model is predicting bud break happening when that solid black line crosses the dashed line. And so using some values that had come from Al's previous work, I was able to build cold hardiness models for my 2 species. The results of this we can see here, again, with observed day of bud break on the x-axis and predicted bud break on the y-axis. And we see that this model is a bit of a better fit than the growing degree day model. The correlation is a bit stronger. And instead of over predicting the day of bud break by 3 days, it's only averaging to be less than a day off. And so for visual clarity, I have the 2 models side by side here. And this makes it clear to see that the cold hardiness model is better at predicting bud break than the growing degree day model. So that brings me to my conclusions, which is, as we have seen, the cold hardiness models were better at predicting bud break than the growing degree day models. And this is super important if we think about climate change with advancing winter temperatures and more erratic and unpredictable shifts in temperatures throughout the year. We want a model that's going to be more holistic and incorporating more of these factors to predict bud break than just simply growing degree days. And moving forward, more work could be done to test the cold hardiness models on more species and in more diverse climatic regimes. So lastly, I just want to say a huge thank you to Al for his leadership and guidance through this whole summer, to Faye for coordinating this wonderful opportunity, to the Harvard Forest for their data sets, and to the DaRin Butz Foundation and the Arnold Arboretum for providing such a really wonderful opportunity. This was probably the best way I could be spending my summer, so thank you.
Thank you, Erica. And next up we have B, who has also been working with Al this summer.
All right. Hi, everyone. My name is Bea Mace, and this summer I worked with Dr. Al Kowaleski to study the effect of environmental cues in floral bud initiation of apple. Particularly, I was looking at the progression of bud development through stages in relation to internal and external factors. So to begin, floral bud initiation in apple consists of 11 stages. It begins with induction, which is when the vegetative bud or adjacent leaves perceive an environmental signal to commit to developing floral structures. Following that, initiation is the progression through physical changes to the meristem as it differentiates into the primordia of the floral organs. The key shift is from vegetative stage 0 to stage 1, shown by a doming of the apical meristem and indicating commitment to floral bud initiation. On the right is the inflorescence that this meristem will develop into the following spring, with one king bloom more fully developed than the lateral blooms around it. The bud then continues to change with the formation of lateral floral meristems, terminal bracts, terminal sepals and lateral bracts, and terminal petals and lateral sepal primordia. Next, the terminal stamen primordia and lateral petals appear along with the hollowing out of the formerly domed apex. Then the lateral stamens, terminal pistils, and lateral pistils form. Finally, all meristematic tissue is fully differentiated into the structures of the terminal and lateral floral buds. So my research is guided by three main questions. First, which environmental signal has the stronger impact on the timing of floral bud initiation, day length or thermal time? Next, how does initiation vary across different shoot structures, and how does it vary across apple genotypes or cultivars? My project is based on a data set collected by Dr. Poliana Francescato over the course of 3 years in Brazil. Developing buds were collected from adult apple plants of the cultivars Gala and Fuji, and from 3 different fruiting structures. The buds were dissected and examined for development to floral structures to create the scale of development I just described. I took this data and made models which fit the data to different explanatory variables, and then I compared across the 2 cultivars and 3 structures. So why study apple? Apple is a globally important crop for which there is much existing research, and it's also a unique study system for bud development because of the different types of structures upon which floral buds can form. The small spur shoot forms flowers in its terminal bud, and the longer bore shoot forms flowers in its terminal and axillary buds. Also, the 2 cultivars of interest exhibit distinct phenotypes for fruiting pattern. Fuji follows an alternating pattern of high and low crop loads each growing season, which is known as alternate bearing, while Gala fruits more consistently each year. In non-agricultural settings, this alternating pattern is also called massed seeding. So getting into the questions, first, which environmental signal leads to initiation? I'll start by explaining my predictor variables. Firstly, days after full bloom is the number of days from full bloom in late September or early October to the date initiation is observed. Days is simply the number of days since the beginning of the growing year, which I define to be July 1st rather than January 1st, since the data is from the Southern Hemisphere. I also looked at growing degree days, and you can recall Erica's great explanation of those a few minutes ago. And that is a measure of heat accumulation based on the daily temperature above a specific base temperature, here being 0 degrees Celsius. I measured the data against GDD accumulated after full bloom and also from the beginning of the growing year. So modeling each stage of development against each explanatory variable, we have days after full bloom, days, GDD after full bloom at 4 base temperatures, and cumulative GDD. The models with the highest residual error values, meaning difference between the points and model line, were ruled out, which are days and cumulative GDD. Looking at what's left, the best fit is for the models based on days after full bloom, with GDD base 0 after full bloom being the next best fit. Graphing the progression of bud initiation by days after full bloom, each cultivar has a specific number of days at which 10, 50, and 90% of buds have reached a particular stage. Graphed a different way, you can clearly see the difference between these milestones for the 2 cultivars. And here are all of the stages, and in each stage, Fuji initiates considerably earlier than Gala. So secondly, how does floral bud initiation vary across the different shoot structures? Focusing on just Fuji, there is an order of progression through the stages, which starts with the spur shoot buds, followed by Boris axillary, then Boris terminal buds. But as we progress through the stages, the axillary and terminal buds of the Boris shoot begin to converge in timing, and from stage 5 onward, there is no significant difference in timing between those 2 bud types. For Gala, the same thing happens starting at stage 6A. Lastly, what differences are there in the timing of floral bud initiation in the different cultivars? So we saw before that Fuji initiates earlier than Gala for each stage, and this is also true across each individual structure, axillary, spur, and terminal. Narrowing our focus to just the Boris terminal buds, I modeled each stage. As you can see, Fuji stays earlier than Gala for every stage. And this pattern holds for spur and axillary buds as well. Finally, I just want to summarize my conclusions and mention some questions that these results brought up for me. Firstly, I found that days after full bloom is the best predictor of initiation, which leads me to wonder what additional variables such as solar radiation might be a predictor of both full bloom and initiation timing. Secondly, initiation begins first in the spur shoots and then in the bore shoots. And I wonder how the chemical signal to induce and thus initiate development travels through the plant and when it reaches the different shoot types. Lastly, initiation began earlier for Fuji than for Gala, and I mentioned earlier that difference in fruit bearing pattern between the two cultivars. And so I wonder if this earlier initiation for Fuji impacts that fruit set and fruit bearing pattern that is observed in that cultivar. So finally, I just want to give a few thanks to people who helped me during this internship. First to Al for all of his guidance and help through this project, to Faye for her support and for leading such a great program, to Ellie for spending a lot of time with me in the lab these past couple weeks, including to take this picture, and to DaRin, Butz Foundation, and Arnold Arboretum. So thanks for listening.
Thank you, Bee. Next up, we have Anisha, and she's been working in the Taylor lab this summer.
Hi, everyone. My name is Anisha Pomik. I use the she, her pronouns, and I was in the Taylor lab this summer studying shrub expansion in the Arctic tundra, specifically using remote sensing tools and techniques. So I wanted to start off by talking about why the Arctic tundra is so important to study. And I think this graphic is really telling. So as you can see here, this shows mean air temperature change over the last 60 years. And the Arctic at the top just lights up in the darkest red. It's experiencing warming at a rate 2 times the global average. And this is even more significant because there's 1,400 gigatons of carbon stored away in tundra soils and in tundra permafrost. So it's really important that we understand what's happening to these tundra soils to better predict and model climate change and understand global carbon cycling. So to give a bit more context, here's a map of the circumpolar Arctic. My study site focuses on a part of northern Alaska. Now, I'm sure when I'm saying Arctic tundra, most of you are imagining a landscape like this. And this is true, this is winter in northern Alaska. However, it's important to remember that in the peak summer season, there's a lot of vegetation and plant life that play a really important role on soil carbon dynamics and nutrient cycling. So one of the key players in this landscape are tall shrubs. So these tall shrubs include willow species and a species of alder that are native to the Arctic. And researchers have noticed through both satellite imagery as well as field research that these shrubs are expanding across the Arctic as climate change progresses. Now, why might this be important? Well, the structure of these shrubs actually have shown to increase the soil temperature near them, and that increase in temperature leads to more microbial activity, which release carbon dioxide and methane into the environment. Now, it's important to remember these shrubs, they're woody plants and they can photosynthesize, so they can also trap carbon. So there's these complex feedback loops at work. And to better understand them, we have to take a closer look at these tall shrubs and ask ourselves, how are they expanding? Why are they expanding? And what the mechanisms may be. So on this question of shrub expansion, researchers have noticed that these tall shrubs are expanding near rivers. These two photos were taken 50 years apart, and as you can see, the shrubs expand right by the riverbed. Other researchers have had similar observations in their field work, but this trend can also be captured using remote sensing tools. So here is a remote sensing derived shrub above ground biomass map. And these are the types of tools that I used in my project to answer my research question, which is, what abiotic factors associated with these Arctic rivers explain the tall shrub biomass that's near them? And to start answering this question, I looked at a few predictor variables. These include soil moisture, slope, and distance to the river. And all of these continuous variables were collected using remote sensing data from NASA and were visualized and sampled using QGIS software. So these were just the continuous predictor variables, but I also developed a categorical variable called terrain type, where I used RGB satellite imagery to develop 6 categories of terrain. So now I have my predictor variables. What am I trying to predict? And that's my response variable, shrub above ground biomass. This is a map of shrub above ground biomass. This has also been derived from remote sensing tools. My process here was then taking all my predictor variables and creating an extensive list of hypotheses, trying to understand how these predictor variables might explain shrub above ground biomass. And to do that, I took a closer look at the biological significance of these predictor variables. So for example, soil moisture might set certain conditions for shrub growth, whereas distance to river and slope might indicate a certain level of exposed mineral soil, which promotes shrub growth. And terrain type might indicate a certain soil type that shrubs prefer. So all of these hypotheses were then converted into models that I tested in a multiple regression framework. Now, my result from all this testing of these models was that soil moisture and terrain type best predicted shrub above ground biomass. We can take a closer look at this data as well. So here is a graph with soil moisture on the x-axis and shrub above ground biomass on the y-axis. The first thing you might notice is that for all these lines, as soil moisture increases, shrub above ground biomass also increases. Now, if you take a closer look at these lines, and these colors are different terrain types, and you can see that these different levels, these different terrains indicate different levels of shrub above ground biomass. So for example, here, valley and hill, for the same soil moisture, will have a much higher level of shrub above ground biomass than the terrain type of gravel bar. To look at the strength of this model, we can compare the predicted shrub above ground biomass that we get from my model versus the observed shrub above ground biomass that we get from the remote sensing data. This dotted line here is the one-to-one line, and if all the data were to fit perfectly, it would be on this line. And this red line shows the linear fit through my points. So as you can see, for lower amounts of shrub, the model does a better job of predicting observed shrub above ground biomass. But as it increases, it underpredicts the amount of shrub above ground biomass. However, this gray shading shows the 95% confidence interval, and it includes this dotted line. So the model is doing a pretty good job of predicting shrub biomass. What can we do with these results? Well, first we can conclude that higher soil moisture is correlated with increased shrub growth near rivers. Secondly, terrain type can help predict the magnitude of shrub biomass, and together, these two variables capture aspects of soil conditions that promote shrub growth and can be used to roughly predict shrub above ground biomass near rivers. In a broader context, my research shows that remote sensing can be used to understand shrub expansion in the Arctic. And the other variables that I use may be poor predictors, but there might be other variables that can play an important role and contribute to my model in a different way. Finally, my result of soil moisture and terrain type serves off as a jumping off point for understanding the mechanisms for tall shrub growth. We can ask questions like, why does higher soil moisture lead to more shrub growth? Or what is it about terrain type that indicates shrub growth? And I would like to conclude with a series of thank yous to Calvin and Ben, who are my mentors and guided me through this project, for Faye for putting on such a great internship and for being so welcoming, as well as all the other DRB interns who have been so supportive and great to get to know. And the DaRin Butz Foundation and the Arnold Arboretum for funding and hosting this program, and for NASA's Above campaign for providing all the remote sensing data. And thank you for listening.
Thank you, Anisha. Next up, we have Millie. She's also in the Taylor Lab this summer.
Hello, welcome to my talk on older machine learning in the Arctic. So, why older? Why machine learning? So older is a nitrogen fixing tall shrub, which has quite a large habitable climate, making a really good pioneer species in a rapidly warming Arctic. Nitrogen fixing is an especially important part of this, as Arctic plant communities are traditionally nutrient limited, and so most non-specialized plants cannot survive there. However, because alder can fix nitrogen, the nitrogen then enters into soils and creates more favorable conditions for new plant types and communities to emerge. However, physical access to plant communities in the Arctic is quite difficult due to ice and snow cover, but satellite imagery has been used to overcome this, as it does not require you to be physically present to collect the data. So as you can see here, this can enable really widespread data collection for variables such as like how much vegetation is in a particular location. However, as you can also see in this image, you can't tell all another tall shrubs apart. You can see here, this is all just green. It doesn't particularly tell you either way which is which. And so sort of my question was looking at how can we differentiate between these species? So that led me to looking at shoulder season imagery, specifically from about September to November. So as time goes on, leaf color changes. And so in this, in the September to October shoulder season portion of the year, you can see here, alder is green, Betulina is this deep red here, and Salix purpura is yellow. So you can visually see what is older and what is not. In terms of the machine learning aspect of this project, machine learning is being used to classify the differences between older and other tall shrubs. The time and effort it would take an individual to hand classify everything would just be immense. Machine learning can do it far, far quicker than a human ever could. And so this can then be built up to include lots of other different factors. And so machine learning has also been used across a variety of fields and has been successfully deployed in a similar shrub ecosystem in the Afar Plains in Ethiopia. So my question is, does a random forest algorithm categorize alder distribution across the Alaskan Arctic well? So in terms of how I actually went about this, you can see here my study site, which is Sagwan Bluffs, and I hand drew the classification polygons here. So down here is where I selected alder, and then I created a load of point data. So I added in spatial coordinates to that to then tell the algorithm actually, okay, here in the space is where this point is located. I then moved the output of these point coordinates into RStudio where we ran the model. So how does random forest work? So I want you to imagine you're in the Arboretum and you're walking from Weld Hill to Honeywell. And so you've got 7 other people with you, you're all wearing different colored t-shirts, and you all follow different paths through the trees. And every time you come across a tree, it looks at you and decides, okay, what color t-shirt are you wearing? And as the people move through the Arboretum, every single tree that you cross makes a decision about what color t-shirt you are wearing. And so once everyone has reached the Honeywell building, all the trees come together to decide the paths that each individual took through the Arboretum, and that's sort of how random forest algorithms work. In terms of results, oh, there we go. Okay, it froze. So in terms of results, we've got a very nice output here on the right, which came out of the model. And so you can see here, it classified various different points of interest. We're mostly interested in alder, which is down here, and here, which is the dark black. And so the accuracy rate for the model is about sort of 0.998, with a confidence interval about 99%, suggesting this is a very good model fit for the data and for also understanding the difference between alder and other aspects of this image that I just sort of talked about. However, this is just the model output, and so this just shows very nicely how the model did in comparison to the data that we gave it. And so over here on the left, we have some validation that I did. So here you can see some ground truth points. And so what I did was I took the ground truth points and took the just the older aspect of the model results and asked QGIS to tell me where they intersected to understand how well the model performed. Obviously, for this particular bit here, which is this particular bit here, it worked really well, which is really encouraging. And so obviously the ground truth data was only across a small area, so we can't compare the entire thing as is here, but it was really encouraging to see. So obviously it performed really well, which was really encouraging. And in terms of why it works so well, it's likely because random forest is actually
designed to perform image classifications. That was its original role. And so the methods that work under the hood of the model really enabled us to get to those finer details about sort of deciding between areas which might be sort of majority older but not all, versus patches that were maybe a little bit of tall shrub, which was really, really useful. In terms of comparing to other studies, Chivarot et al. Used a much more complex model with far more inputs in the Afar region, but the results are very much in line with theirs, suggesting that potential refinement of this model could really yield a lot more information about how we can sort of start to really understand older locations across the Alaskan Arctic. So in terms of my final thoughts, this is progress both for the field of remote sensing in the Arctic and for understanding machine learning classification in general, which is really exciting, and obviously it does work. There is the potential for much better and much more refined models to predict much, much larger scale data sets, which can also really help sort of understand how the Arctic is changing in terms of tall shrub expansion. And then there's even the possibilities of moving on from tall shrub expansion currently to predicting where there might be favourable conditions for future shrub expansion. And finally, I'd just like to say thank you very much to Faye, Ben and Calvin for all their help so far on the internship. This has been such an amazing opportunity. And then I'd like to thank the Arboretum, Durham, Hillbead and the DaRin Butz Foundation for enabling me to take up this opportunity. And please feel free to ask any questions about any of the presentations that you've seen so far today. Thank you.
Thank you, Millie. So now we have seen all of our presentations except for one of our intern who is unfortunately in Guatemala right now, but he will be at the Arboretum in 2 weeks' time, where he'll be able to visit with everybody and then present his presentation then. So I hope you all come for there. But now you have a whole field here. If anyone has any questions, please feel free to ask them. And I can't really see you, so just unmute yourself and ask. Any questions? Maybe not. All right then. I guess it's time for ice cream and sandwiches, so we will see you all outside and you can ask any more questions you might have out there. Thank you very much, everyone. Oh, yeah.
This is Ned, and I just need to say a couple of words. To all of the DaRin Butz research interns, under trying circumstances and the complexities that we're living with now, they were all fantastic presentations. We're so glad to have had you here. And Faye, I want to say something to you. Your leadership and the support of the DaRin Butz Foundation is helping to continue to launch the careers of the next generation of scientists. Faye, your efforts have been heroic, and I just cannot thank you enough. All right, let's get some ice cream and some sandwiches, or the other way around.